Hello, my name is Saiful Ikmar bin Ahmad Azhar. My metric number is F20A0589. To understand farm animal perception and respond to environmental stimuli as behavioral, it is important to reveal neural link between sensing and behavioral output in the brain. As animals are taught to categorize whether sensory stimuli are positive, rewarding, or negative aversive. The study of rewarding or aversive neuronal mechanism provides us with possible criteria by which to determine the manner of husbandry preferred by farm animals. Rewarding or aversive response are reflected in the functional chain of the midbrain, which is known to cause emotional behaviors. Therefore, study about neuronal mechanism underlying learning and memory are required in order to understand behavioral development in farm animals. In addition to interest in behavior, stress has been a large area of concern in the study of farm animals. Behavior is often a good index by which to know whether the animal have become stressed or not.
Hearing. In cattle and goats, the hearing modality was examined by changing in the position of the sound source. The source is changed along the medial sagittal plane. 18 to 30 degrees is the threshold for showing behavioral responses. In human and dogs, the threshold is 0.8 and 8 degrees. The finding indicates the cattle and dog and goat are less sensitive to the change of the sound source than human and dogs. Similar results were obtained in pig. I will present about taste. In condition taste vision, the animal first receive the conditioning and an injection of lithium chloride together. Once the animal takes the combination of conditioning with lithium, it refuses certain kind of the task. Third, the conditioned taste aversion is used for training cattle in the field not to eat toxic grass. The taste aversion learning task occur even when the cue and the aversive experience are separate for many hours. However, the neural mechanism underlying a specific association between cue and aversive experience in the central nervous system is unclear.
Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Taufik bin Ismail. My number metric is F20A0475. Today, I want to present about olfaction. So, olfaction is one of animal key rules to recognize and confirm objects. After penetration, mitral cell of the olfactory bulb will respond to the lamp odor that has been demonstrated by Kedrick et al. So, gamma-aminobutyric acid and glutamate will release only in response to the odor of the sheep on lamp. Meanwhile, acetylcholine and norepinephrine do not have any specific response. After penetration, the odor discriminating mechanism of the lamp will activated in the process from the centrifugal input to the olfactory bulb. Olfactory discriminating ability of the lamp weak in the case of cesarean operation.
My name is Muhammad Chalil Zahar and my metric number is F20A0671. I would like to talk about a study about vision. Using 2 Holstein Friesian and 1 Red Holstein Bulls, Kemper and Goreh in 1998 report that bulls could identify a white center as small as 1 cm of a black disc. Bulls have a visual angle of 23 arc minutes, but this acuity value is smaller than that of sheep and that of younger cattle. These findings suggest that the vision acuity of the bull decreased with aging. Next is further test by Kemper and others in 2000, a hypothesis using adult cattle that visual perception in oval pupil in the eyes is different for stimuli with vertically separate detail rather than stimuli with horizontally separate details. When the bull receives the task, which is the operant conditioning task for discriminating a solid white line from a white line with decreasing interspace, it solved the task better with the vertical stimuli than the horizontal one. Next is study has been done by Kendrick in 1991 in sheep about their ability to visually recognize human. This study clearly shows that the cell activity in the temporal cortex of sheep exists, which respond to face or bodies of human. Last is in recent study by Prot and others in 2000. It was found that exposure to upright face selectively increased C-force RNA expression more significantly in the right temporal cortex than in the left. The brain of sheep may have the neuronal circuit which discriminates upright face in the right temporal cortex, as do human.
Assalamualaikum. My name is Nasrul Hafizan bin Muhammad. My match number is F20A0482. I will tell you about amygdala. Many previous studies have shown that amygdala play an important role in the expression of autonomic and behavioral change when the animal receives stressful. Stereotactic studies have demonstrated that ablation complex block the responses induced by stressor and that the central amygdaloid nucleus is a part of neuronal circuit mediation activity. The recent study suggests that part amygdala, lateral amygdala, is a likely site of the plasticity underlying memory storage for fearful conditioning. Thank you.
Next, we will look into the autonomic nervous system. In autonomic nervous system, it is consists of three neurons, which is the sympathetic neurons, the parasympathetic neurons, and as well as the enteric neurons. And all of these three neurons' activity is largely influenced by the amygdala and the hypothalamus. And it is well known that the heart rate, the blood pressure, and the hormonal secretion from the adrenal glands are strongly regulated from the autonomic nervous system. With regards to the heart rate, domestic animals can be grouped into the parasympathetic dominant, for example horse, cow, and goat, and the sympathetic dominant animals, for example dog, pig, and chicken. The regulatory mechanisms of heartbeat by nuclei of amygdala and hypothalamus may differ in the sympathetic dominant from that in the parasympathetic dominant group. Analysis of changes in the rhythm of heartbeat or blood pressure using analytical programs such as the fast Fourier transform algorithm, the wavelet, or the autocorrelation program is useful to know the reciprocal neuronal innervation of the autonomic nervous system.
Assalamualaikum. I am Muhammad Shahni bin Zulkifli. My metric number is F20A0473. I will talk about central regulations and stress-induced behavior. Corticotropin-releasing hormone (CRH) is a key hormone integrating the stress response in the central nervous system. ICV processing CRH decrease the oral or nasal stereotypical behaviors, road and leak in the pit. Pigs given cortisol had no apparent change in behavior. ICV CRH may differently act on the neural pathways regulating behaviors and vocalization from that of endocrine responses. ICV CRH may have longer effects on the neuronal network that controls behaviors in the pigs. Similar effects of the central CRH may be present in cattle that are exposure to stressors rapidly increased adrenocorticotrophin, ACTH, and corticosterone cortisol secretion. Based on open file test plasma ACTH levels from orally tethered curves where high stereotypes are generated from animals that are not all low stereotypes, and that curves with high stereotypes indicate more exploratory behavior. Radbo in 1998 stated ACTH levels in curve are much higher than in the heifers, but cortisol secretion after exogenous ACTH administration is lower in curve than in heifers. This study shows stereotype behavior may occur in curve or heifers with low plasma ACTH levels. Concerning separation and reunion in cattle, Berzi and Liniandre in 1997 provide one model for investigating the psychobiological basis of social attachment. During short-term isolation and to subsequent reunion with familiar or unfamiliar conspecified behavioral heart rate and plasma cortisol were measured in the upright and Parisian cattle. According to Shader and Ledwin in 1999, recurrent social isolation in pig caused a persistent decrease in pituitary adrenocortical axis response, but epinephrine levels, activity, behavior, and heart rate remain unchanged. This suggests a separation of the pituitary adrenal response from behavior and other sympathetic activities. The cortisol level is often used to assess chronic stressful situations in the animals. Dechon in 2000 showed that barren-house pigs at the age of 22 weeks of age had a blunted circadian rhythm in salivary cortisol concentration compared to enriched house pigs. That's all from me. Thank you.
Assalamualaikum warahmatullahi wabarakatuh. Today I would like to discuss about the stereotactic surgery. My name is Siti Nur Shakira binti Ishak. My number matric is F20A0609. Okay, it is widely accepted that the brain integrates the neural networks that control the sensory function and the behavior outputs, and that neural nuclei of the hypothalamus and limbic system play a key role in regulating motivates, emotional behavior, and memory. To analyze the regulatory roles of an intercerebral nucleus in some of the sesame regulations. A stereotactic instrument and the brain atlas are required. The coordinate for the integral structure are calculated from the three standard planes, sagittal, corona, and horizontal, with the axis of the stereotactic instrument horizontal. With the edge of the stereotactic instrument, skull structures, and the size of the brain differ largely among animals. Therefore, the instrument has been designed and made for each target animals. In the domestic piece, Machilus ETL 1990 first provide the brain atlas of the landless pipe weighing about the 40 kg. They indicate in the paper that visualize of the interbred market is useful for making the atlas. Cited ETL 1998, however, show an alternative way of making the atlas of P where about coordinate matters is able to be set based on the external school structure. The instrument for larger animal has not been established, but a prototype of the instrument for cattle was recently developed. Sato et al. 2001.